All right, let's get started. Today, we're putting on our detective hats. But our crime scene? It's nature itself. And we're hunting for clues to find the vast majority of life that's totally invisible to the naked eye. We're about to uncover how scientists are solving this massive mystery, so let's follow the evidence.
I mean, really think about that for a second. It's the biggest challenge for anyone trying to figure out what's actually going on in an ecosystem, right? The old school ways, they rely on what we can see, or what we can catch, or what we can hear. But what about the creatures that are super elusive, or microscopic, or just so incredibly rare you'd never spot them? Well, this is exactly where our detective story kicks off.
So here's the big secret. Imagine every single animal, every plant, every fungus is just constantly shedding these tiny invisible pieces of itself. We're talking skin cells, mucus, waste, pollen, you name it. These are what we call genetic ghosts, a trail of DNA that gets left behind in the water, in the soil, even floating in the air. And for a genetic detective, these ghosts, they are the ultimate clue.
But, before we dive into how we actually track down these genetic ghosts, let's talk about why we even needed a new playbook. For a long time, our options were pretty limited, and frankly, they came with some serious problems.
Just think it through. Trying to physically capture animals with nets or traps, it can be painfully slow and takes a ton of work. Not to mention, it can really stress out the animals and sometimes even damage the very habitat you're trying to protect. Plus, you're almost guaranteed to miss stuff, especially the small, well-hidden or nocturnal species. We really needed a better, less invasive way to see what's out there.
And that is where this revolutionary forensic toolkit comes into play. Scientists can now analyze those genetic ghosts we were just talking about, using an absolutely game-changing technology called environmental DNA, or as you'll hear it called, eDNA.
So eDNA is basically the collection of all those microscopic genetic fingerprints. The skin cells, the waste, all the little bits that organisms can't help but leave behind. Instead of spending weeks trying to find the creature itself, we're just looking for the clues it left at the scene.
And if eDNA is the clue, then you can think of meta-barcoding as the high-tech forensic lab that cracks the case. It lets us take a single sample, say a bottle of pond water, which is kind of like dusting the entire crime scene for prints, and it tells us every single species that was there, all at once. It's a massive leap forward.
Okay, a quick but really important distinction here, because you might hear this other term floating around, community DNA. That's a bit different. With community DNA, you actually have to go out and collect a whole bunch of organisms first, like scooping up insects in a trap, and then you basically put them in a blender to get their combined DNA. But with eDNA, you get to skip capturing the critters altogether. You just grab a sample of the environment, the water, the soil, and you pull the DNA directly from that. Much simpler, right?
So how exactly do we go from a scoop of muddy pond water to a complete list of all the fish and frogs and insects that call that pond home? Well, the process is actually surprisingly straightforward. Let's walk through the workflow together.
It's really just a three-step process. First, you're out in the field and you collect your sample, a bottle of water, a little bag of soil. Simple. Second, you take it to the lab. You pull out all the DNA and then you make millions and millions of copies of it using a process called PCR. It's basically a genetic photocopier, and it makes sure you have enough material to analyze. And third, you're at the keyboard. Powerful software sifts through millions of these DNA sequences, matches them to a huge reference library, and bam, it spits out a species list. You go from a muddy sample to this crystal clear picture of biodiversity.
Okay, so we've got the technique down, but what can it actually tell us? What kinds of secrets are these genetic clues revealing about the world all around us? Honestly, the results are pretty mind-blowing.
When we put eDNA head-to-head with the old-school methods, the results are just incredible. Study after study finds that a simple water sample can turn up more amphibian species than weeks of traditional surveys. But the truly wild discoveries come from the most unexpected places. Get this. In one study, scientists analyzed the blood inside of leeches they collected from the forest floor. And from the DNA in that blood, they identified endangered and super elusive mammals that even high-tech camera traps had completely missed. It's the perfect example of how this tool helps us find that hidden majority.
It's an incredibly powerful tool, there is no doubt about that. But, just like any good detective, we have to understand the limits of our evidence. Finding a suspect's DNA at a crime scene, that proves they were there. But does it tell us what they were doing? Or maybe more importantly, how many of them there were?
And this is what's known as the abundance challenge. While eDNA is absolutely fantastic for telling us who is in an ecosystem, it's a lot trickier to say how many of them there are.
For instance, the little chemical primers we use in the lab to find DNA might just be better at latching onto the DNA from one species over another. This can kind of skew the results, making it seem like a species that sheds a lot of DNA is way more common than it actually is. So for now, its real strength is presence or absence, not doing a precise head count.
But here's another really cool thing. The story the DNA tells also depends on where you find it. You can think of DNA as a kind of time machine. If you take a sample of honey, it can reveal exactly which plants a bee visited locally over the past few days. But what if you drill a sediment core from the bottom of a lake? Whoa! That can reveal the entire history of the plants and animals in that whole region, stretching back for thousands of years. We're literally reading clues from both right now and the deep, deep past.
So, we have this incredible new detective tool that can read clues from both the past and the present. But why does that really matter? What's the real-world impact of being able to survey biodiversity this efficiently?
Well, first up, conservation. This is huge. We can now monitor some of the world's rarest and most threatened species without ever having to see them, touch them, or disturb them. Just a scoop of water can confirm if an endangered frog is still hanging on in some remote pond, which lets us launch rapid, targeted protection efforts before it's too late.
Second, it is a total game changer for catching invasive species. We can use eDNA for what's called passive surveillance, just constantly monitoring pathways like the ballast water in huge cargo ships. This means we can get an incredibly early warning, finding an invasive species way before it has a chance to take hold and cause millions of dollars in economic and ecological damage. It's about stopping the problem before it starts.
And finally, and this might be one of the coolest parts, it puts this incredible power into way more hands. The fact that collecting a sample is so simple means that pretty much anyone, from school groups to local communities, can take part in these massive biodiversity projects. It empowers citizen scientists to become the detectives monitoring the health of their own backyards, creating this amazing, unprecedented global network of environmental data.
You know, when you step back, we've basically unlocked a completely new way of seeing the world. Environmental DNA lets us read the biological history and the present-day reality that's written into the environment itself. It's non-invasive, it's scalable, and it's unbelievably powerful. We've really just opened the first few pages of this immense living library. So the real question is, what other incredible stories are just waiting for us to discover?